And by 1837, I think, yep, he was already starting to think about the fact that species were not fixed like he had been told. This is the famous "I think" stick diagram that he wrote in his notebook B in 1837, and then in 1838 he read Malthus, and the pieces all kind of came together. So let's move right on. Thank you. And so, why should you even care about evolution? You're majoring in history or art or music, and how does evolution affect you at all? And I think it's because mainly we all have an origins worldview. And some people who are religious get kind of an off-the-shelf variety, and everyone's got the same worldview with the same answers to questions. And for the non-believer, we have to put our own worldviews together. And most people who are non-believers choose secular humanism as a worldview. So from these worldviews, we just derive our meaning, purpose, our moral values, what standards we use, and also, more importantly, how we view reality and how we find truth. The beliefs are often held so closely that, especially in believers, that these are elevated to sacred values. They become part of the core identity. And so if you're in a discussion with someone about origins and that affect their beliefs, it can actually feel like an attack on them if it looks like what they believed isn't true. So origins form a foundation to our worldviews, basically, is what I'm saying. And surprisingly, theologians, philosophers for thousands of years have tried to address where we came from, where humans came from, what it means to be human, all the existential main questions that philosophy and theology especially address. And then along comes science, 400, 500 years ago, and not wanting to answer those questions. It's basically just dealing with nature. But when you know where humans came from, when they arrived here, how they got here, and those questions, as you begin to answer those with facts of nature, they inform the why. And so, in my opinion, science began answering indirectly existential questions that they weren't really looking for, and many times conflicted with what previous people said. So what excites me about this, and why you need to know this, what about evolution? How does it affect you? Well, it answers a lot of the existential questions that people have been pondering about for thousands of years. So, and obviously it also falsifies a lot of other competing narratives about where we came from and how we got here, when, etc. So I would ask you, if you had to think of what's the most important discovery in mankind or humankind, people can think about fire, that we tamed to use for ourselves, and perhaps Copernicus and the realization of how the solar system was originated, or maybe Newton or Einstein. But I would assert that actually the most incredible discovery made ever is evolution, because it affects everything. If you're a music major or you're bringing out emotion out of people, or art, or humanities, looking at what makes a human, how we deal with our relationships, etc., none of that can be addressed completely and with understanding without an evolutionary context. And so I believe, since evolution affects literally almost every part of our lives, it's the most important discovery in history. So my main purpose today is say, okay, so how do we know that evolution is true? And I'm just going to settle on two examples. I've been at this for 40 years, debating all kinds of people about evolution, and I think I've heard just about everything. But now I think there's a couple of great examples out there that are holding up against anti-evolutionists and people who want to dismiss evolution as being true and important. And so I'm going to go through those today. But there's a website that I've just established a couple months ago, and the two things I'm going to be talking about today are on the website, which you see at the bottom, truthfulorigins.info. And so don't worry about trying to understand everything today, because you can go to that. There's also blog entries on there on homosexuality, abortion, morality, where it comes from, how we can study it, et cetera. So they're my opinions, but they're really not. I think basically they're consensus of the scientific community about a lot of things that I think you might be interested in. So don't worry about, sit back, let's go through these. And if you get lost along the way, you've got that website to go to that will, I think, for just a couple minutes reading per topic, tell you what's going on. Okay, let's go. Let's jump into things. And when you look at why somebody might ask, how do we know evolution is true? I think it's important to find out who's asking and why. Is it a biology student, a creationist who just wants to find out why you're wrong? Is it a believer, maybe that's just interested? Is it an apologist? If you're apologetics is defined as the defense of the faith, not the defense of the truth. They think that it's the same. But if you're discussing evolution with an apologist, my experience has been is that they're not wanting to know why they're wrong. They're wanting to have you educate them into the latest scientific facts so that they can come up with more reasons why you're wrong. And so as long as you realize, if you're in a discussion or a debate or a dialogue with an apologist, there's two different scenarios going on here, and it's not the same. If you're looking for truth or why you could be wrong, they're not. They're finding out why you're wrong and where they need to read. So maybe it's an engineer who has no biology background. Many of the young Earth creationists are engineers. Maybe it's a medical provider and they're going into medicine for reasons that they want to be on the mission field, et cetera, and they don't believe evolution. I just finished a blog a few days ago on why evolution is important to the medical field. And so you might want to, if you have an interest in that, read that. So even if you're a humanities, history, or artist, music, sculpture, the theory of evolution does touch you in terms of meaning, purpose, and what it means to be a human. It has to be in an evolutionary context. Okay, let's go to the next slide. All right, so where do you go to find out why we know evolution is true? And with the internet now, there's books, there's movies, there's podcasts, great video series. I've put a lot of these on my site under resources. And by the way, I'm sorry I'm putting my site up as where to go. I don't get any money from it. I just think it's a great resource, consensus. It'll save you a lot of time. But there's various fields of biology that if a person has an interest in biogeography, which is how Darwin kind of came up with his Galapagos finches and the various, the tortoises, there's paleontology, fossils, transitional fossils, et cetera, developmental biology, DNA is incredible right now, what we're finding out with evidence for evolution. I'll be talking about that. Comparative genomics using haplotypes and kind of nested Russian dolls, we can trace back when humans left Africa to colonize the world. And the most successful exit out of Africa happened about 70,000 years ago. And to our surprise, using genetics, they did not go into Europe or even to Eurasia. They followed the coastline and ended up in, of all places, Australia. So just really good, incredible evidence and facts that were coming out. There's vestigial structures you can study. Faunal succession refers to the fossils that are found in different layers, uniquely going from simple to complex. So, and then there's a radiometric dating. I have a quote down here. I'll let you read that or come back to it at another time. But basically, Theodosius Damasky was a very famous biologist who also happened to be a Christian. And as he states down there, that anything suggesting that there is a scientific reasons to doubt the facticity of evolution would be propagating a plain untruth. And so we know evolution is both theory and it's also fact, just like germ and cell relativity theories are also fact. Okay, let's go on to the next slide. All right, some housekeeping here real quick. Evolution is defined as the change of alleles or genetic components of a population over time. So basically, individuals do not evolve, but populations do if there's certain things for selecting which genes are better in the environment. And the main mechanism or a mechanism for that is natural selection. And as you may recall, the title of Darwin's abstract, he called it. It wasn't really the book he wanted to do, but he had to rush it into production because of Wallace. Was on the origin of species by natural selection. So he was mainly proposing a way that evolution could come about. There were other people that were seeing that evolution was probably happening and had happened before him, but he was the one who came up with a proposed mechanism that would actually produce it. And then, as you know, Darwin's proposal for natural selection was read. He wasn't present in the Linnaean Society in 1859 or 58. His book was published in 59. So I think he was proposed in the year before. And Wallace wasn't present either. He was still in the field in Indonesia. So Darwin and Wallace both proposed natural selection as a way that species evolved and populations changed. The other important definition I'm going to get through here is vestigial structures. So vestigial structure is not something that's necessarily without function. It could be without function, but it also includes a structure that has lost its original function and has a new one now. And so I've got some pushback from people saying that is a new way that evolutionists are defining that to get out of problems. But in my slide and video presentation on whale evolution, I can show you that that definition actually goes back to the 1960s, 60 years ago, that it's not just a structure without function, but it's also a structure that has lost its original function. Micro and macroevolution, those usually refer to changes, microevolution at the species level, and then macroevolution is above that level. By the way, I didn't give you a good definition for natural selection, but it is basically differential reproductive success among individuals within a population. And Forrest Valkyrie, which is a YouTuber and science educator, defines it as a non-random selection of random mutations. So again, a mechanism. And then the other thing that didn't show up on this slide is science and religions go about seeking truths in a different way. And religions basically come up with a scenario and then they try to support it. And that's how we end up with thousands of religions and thousands of gods. And they kind of do battle among themselves. Literally, people have been killed over this, where science takes the other approach. And so it comes up with a hypothesis or theory, and it tries to disprove them and only keep the ones that are holding up. And so one way of seeking truths leads to a consensus and hopefully onto the one or two things that are true. And the other one, if you're going to try and prove something but not try and disprove it, you'll end up with all kinds of variations and nowhere near the truth. So let's go on to the next slide. Okay, so I told you I have two examples that I think are holding up great in terms of if somebody wants to know, give me your best example of evidence for evolution. The first one is whale evolution. And I'm not going to talk about that at this time. I'm just going to give you some tidbits of what you'll see. I made 3 videos because with whale evolution, the nice thing about whale evolution is you can actually see the evidence. And so that's what I've done in my 3 parts that are on my webpage. You can click on them. Each one is 25 minutes long. And I want to show you the evidence. I don't want you just to tell you about it. This summary page, a screenshot here, is everything that is discussed and shown in for whale evolution. And when you see it all, it's overwhelming. But here's some little screenshots. The upper left-hand corner, you can see that the blowhole through fossils is moving up towards the present position it is now with cetaceans. The hand underneath that is actually a river dolphin hand. And the point is that if you're going to design and make a flipper, I don't think you're going to use the same bones that are in your hand. And by the way, that is reversed, unfortunately. That little small digit on on your right side should be on the left. So to flip that. When you dissect a seal flipper out, you can see it looks like a hand. Again, this is an argument from homology. If you're going to design creatures that don't have any evolutionary past, why use the same bones that are in your arm, in a bat's arm, and all the ones from the tetrapods, basically. Down below, you'll see a picture of a fetal baleen whale. The mother was killed, and so this was stillborn. And they cut through the jaw, and what you see is a row of dots there, and those are teeth buds. And so most of you may know that baleen whales don't have teeth. They use a filtering process to eat. And so why would a baleen whale that's never going to have teeth have tooth buds that have dentine and no enamel in the fetal stage? And then to the far right, you can see the development of a dolphin in embryology evidence. And it starts out with what looks like a nose, and then it slowly migrates as it ages to where the blowhole's at the top. So there's DNA evidence, there's fossil evidence, there's all kinds of vestigial evidence from our living cetaceans and whales today. And I think you would enjoy this example here of whale evolution, that when you look at all the evidence that whales have evolved, it is overwhelming. Also on my slides that are on through the web page, my website, they're all, most of them are cited, so you can actually fact check me, and all the original sources are on the slides. But this is really great, especially for those of us who are visual. Please, go ahead and look through my whale evolution videos, which are on the site truthfulorigins.info, and I think you'll enjoy seeing some incredible evidence. However, it's not the best. And let's go to the next slide. And okay, so this involves DNA evidence. And this is the best evidence I've seen in 40 years. And it involves shared endogenous retroviruses. And again, I just want you to understand what's going on here. And if you want to dive into the details some more, it's on the website. So what is happening is that there's, as you know, when you get infected with a cold virus or a lot of other viruses, they get into your cell and they take over the machinery, the ribosomes, to make their own products. And so they can keep reproducing inside your body until your immune system catches up with them and is able to corral them. But there's a group of viruses that are called retroviruses. So instead of having DNA, they have RNA. And what they do is dastardly. They actually insert their genetic instructions into your chromosomes. And so they're like parasites. And because they have RNA, and they have to convert their nucleic acids to DNA before they insert into your DNA, they're called retroviruses, because most organisms go from DNA to RNA to proteins. That's the paradigm of nucleic acid reproduction. But retroviruses go the opposite. So you have these viruses that are inserting their genomes into yours, and now their DNA instructions become part of yours, and we have no way of removing that. And so your body can silence them and dampen them down through methylation and other ways to try and control it. But once they're into your chromosomes, they're basically in there. So when the Human Genome Projects were getting their first drafts out, and they found out that we have 3 billion base pairs and only 20,000 protein producing genes, they also found out that 8% of our 3 billion base pairs are actually made up of these old retroviral insertions. And when the genomes that the retrovirus has put into you, when it gets degraded and can no longer work and produce new viruses, but it's still there, we call them endogenized retroviruses, because they get fixed into the population as people reproduce, if the virus inserted into an egg or a sperm. So when you go down as a scientist and you're reading the genetic language that's in the chromosomes, there is a specific component to these endogenous retroviruses that show up, which you see hopefully on the slides in front of you. And so there are 3 genes to a retrovirus, and they're called GAG, POL, and ENV. The GAG produces proteins that are made for the capsid. It's like a lunar module that has to get into the cell to parasitize it. The POL has a lot of enzymes such as integrase and reverse transcriptase, and the ENV has the envelope genes that constitute a part of the retrovirus. And in a couple minutes, I'm going to show you a 5-minute video from the HHMI, the Howard Hughes Medical Institute, which shows you all this. Now, so there's only 3 major genes, and the virus, when it infects you, it needs to be able to reproduce itself and make new viruses. And unfortunately, it uses your system to copy itself, and your system is not set up to read all of the genome of the virus, the provirus. And so the retrovirus needs to have promoters, which are another set of genetic instructions to get it started to make the new viruses. And so before it inserts its GAG, POL, ENV into your chromosomes, it makes these promoters, huge numbers of promoters, hundreds or thousands of them, and they are called LTRs, for long terminal repeats. And so it makes those, and it packages those with it and sends it into your chromosomes after cutting it into. So what you see in front of you are the 3 major genes, and then on either side, flanking them, are the LTRs, which come from the retrovirus. And the other thing that is there are called target site duplications. And if you look at the retrovirus there, the target site duplications are the ATTAT. That's the sequence on either side. And what that represents is when the retrovirus goes to insert its genome into your chromosomes, it takes like a pair of scissors and it cuts your DNA, pulls it apart, and then it slices the retrovirus genome into yours. And so there's actually like 2 knots on either end of your DNA chromosome section where it was inserted in. Those are the target site duplications, the TSDs. And so when a geneticist reads along your chromosomes, your DNA, they can find these endogenous retroviruses because it has the 2 LTRs, the genes, the GAG, POL, ENV, and they know that you can see where it's cut in. So we know that they were not original to your genes, that they're viral, they're parasitic instructions that are inserted. Now, over millions of years, these get degraded and get mutated and they don't work anymore. And so a lot of the GAG, POL, and the EMV have dropped out, and all that you see is an LTR. And we can get into why that is important for a creationist to talk about, or you can read it on my site. But has everyone got it? You have a parasite like HIV, that sometimes can go to AIDS if it's not treated. HIV is a retrovirus. It inserts its genome into yours, becomes part of yours. You can't get it out. And over millions of years, they degrade down, and we find these in your genomes. And so you're saying, what is the big deal? He's wasting all my time. Well, as we're going to see in a minute, what is the crucial point is that these things, which are tens of thousands of these are in our genome, they insert randomly. Now they have preferences, like they like lines and they like chromosome 19 more than other chromosomes, but the exact address, the ATCG sequence, is random. And so that's number one. And then number two is when you sequence orangutan, gorilla, and chimp genomes, you can find their ERVs and compare them to our ERVs. And in terms of chimps, the astounding fact is that we have 200,000 of these, and they match exactly to the chimp genome. So if they're inserted randomly, and there's 200,000 of them, and they're in the same places, there's only two possibilities. One is that for some reason they all got inserted in both species separately, which is mathematically impossible, or the only rational explanation is they had to have inserted before the chimps and humans split 7 million years ago. It's the only way a random insertion is going to be in the identical points. So I want you to sit back, and we're going to see if we can get this 5-minute video by HHMI going here in a minute. Remember that they're inserting into your DNA. There's an enzyme called reverse transcriptase, which converts their RNA to DNA so it can be inserted. And there is another enzyme called integrase that clips the DNA to put it in, and that the virus takes that in there with them. So go ahead, Matthew, and let me have this for 5 minutes, and we'll sit back and watch.
So this is HIV. It's a typical retrovirus, meaning that it has an outer envelope, and in the center,
go ahead and expand it.
RNA, as well as an enzyme here in blue that's reverse transcriptase, which will ultimately turn that RNA into DNA. The virus itself, with this outer envelope protein, actually directly infects T helper cells. The way that it does this is that as it comes up to the cell surface, it uses receptors that are on T helper cells and exclusive to T helper cells, which are CD4 molecule, which really defines T helper cells. It's a surface receptor that binds to the envelope protein. That causes a conformational change and allows a second receptor to grab hold of the envelope. This is the chemokine co-receptor. It's also called CCR5, and we'll talk about that more. What happens now is that the stalk of the envelope protein pierces through from the virus into the host cell and starts to draw the cell membrane and the viral membrane together. And what ultimately happens is fusion of those two membranes, and the viral genetic material is injected essentially into the cell, and the envelope protein is left at the cell surface. The virus has a matrix and a capsid protein, shown here in green and red, that essentially are digested when it enters into the cell. That releases viral enzymes and the viral RNA. And here we have reverse transcriptase, which takes the viral RNA and, using host nucleotides, converts that viral RNA into a single strand of DNA. While it does that, it makes some random errors, which is characteristic of reverse transcriptase. It has very poor proofreading activity. That single-stranded DNA now is again reverse transcribed into a double-stranded DNA. At that point, another enzyme that has come in with the virus in the beginning, called integrase, essentially grabs hold of that double-stranded DNA and carries it through a nuclear pore into the nucleus of the cell. Within the nucleus of the cell, it finds the host chromosome, and it basically, the integrase enzyme, makes a nick in the host DNA and allows for HIV to insert itself into the host chromosome. And that right there is what establishes lifelong infection. Now, RNA polymerase comes along and makes messenger RNA. Those messenger RNAs encode for different viral proteins. They end up associating with ribosomes at the surface of the rough endoplasmic reticulum. And here's a piece of mRNA that's making envelope protein, which is directly produced into the endoplasmic reticulum. And it's shuttled then through the endoplasmic reticulum and taken to the cell surface, where at the cell surface it becomes embedded in the cellular membrane, and at this point coalescing with other envelope proteins that have been produced. You have this cluster of envelope proteins now on the surface of this infected cell. At the same time, there are other messenger RNAs that are being produced that allow for translation of other viral proteins. So here are additional viral proteins being made, which are going to be used to make up the key components that the virus ultimately is going to need. These are transported again to the cell surface, to the area where these envelope proteins are, and a strand of RNA as well as some of the enzymes are part of that complex. This then buds off at the cell surface at this point, but it's still not a mature virion because the polyprotein chain needs to still be digested into its component parts. That's done by an enzyme called protease. Protease breaks up those polyprotein chains and ultimately allows for them to coalesce and form the mature structures that make up the final virion. And now you have a mature infectious virion that can go on now to infect other cells. Once that happens, now the cell can produce tons of viruses, and this is really what then keeps the whole process going.
Okay, thank you. We'll get back to my slides here in a second. Matt's doing a great job. Oh, there seems to be also one of them missing. So let's go back to the other slide, even though we're not going to play the YouTube video. Okay, so I had a summary slide here. So these are not retroviruses. They were retroviruses, but now they're fossilized genomes. They're basically like a second fossil record for us. So the concept is very simple. These are infections into our ancestors. They were random insertions, 200,000 of them. Some people say 98,000, but they're by the thousands. And so we find the exact same ERV in the same location in chimps and humans. They oftentimes have the same mutations. The only rational explanation is that they had to have inserted before the species split. That's basically the whole part of this. And so you can get into discussions with apologists about, well, are they really random or are they really ERVs and all this stuff? And there's just wonderful. They've done experiments. They've actually resurrected an ERV using old ones. So it didn't do what they said it was going to do. It made new retroviruses. There's no doubt about everything we talked about so far. So again, if you want specifics, great. But we now have, I believe, the firmest evidence for evolution we've ever had. One of the Christians who are theistic evolutionists have stated that this is a Galileo moment, a second Galileo moment for theists around the world, because it is no longer logically possible to deny that humans have evolved. And this is also a great example of macroevolution, because you basically have what people want. They want to see human evolution, but they also want to see large changes. Obviously, my first example, whale evolution, going from an animal, a deer-like animal the size of a raccoon, to a blue whale in about 20 million years. The fact that we can show that is definitely macroevolution. And then this is the second type of macroevolution. So I apologize for those slideshows that didn't work, but I do want to, before we end this presentation on those two examples, whale evolution and shared endogenous retroviruses, let's go to the next slide. And there's other DNA evidence for evolution and human evolution, which I'm not going to talk about now, but it's on my website. There's just a wonderful how we figured out that all the great apes except for us have 48 chromosomes and we have 46. And so if we evolved from a common ancestor with them, the question is, well, what happened to the DNA? Where did it go? And it didn't go anywhere. It just got rearranged. And it turns out, if you look at the left side there, this is a diagram of the banding patterns when you stain chromosomes. And you can see that the H stands for human, the C stands for chimp. And if you line them up, you can see how almost identical they are. They've got the same DNA codes, et cetera. And basically, there's only two general possibilities. One is that they are alike because we are primates, and a common, it's due to common design. The great engineer, when he was making or she was making humans and the other primates, you would expect them to be the same. So it doesn't rule out that possibility. But later on in the year 2000s, they eventually were good enough to actually sequence this. And if you look at the right side, this is the exact point. There's a little tiny white arrow, if you can see it. That is where the two chromosomes fuse together to give us 46 instead of 48. And chromosomes have tips to them. They're called telomeres. And so if you fuse two of them together, you'll end up with telomeres in the middle, which they shouldn't be. They're only found on the ends. And then this is the middle of human chromosome 2 where there's a fusion. So it's still, you could just say that that's how God created humans. They just fused them for some reason. Or you can say, well, with shared endogenous retroviruses and with this fusion, it's all beginning to add up into a huge snowball going downhill for evolution, and common design as an explanation why there's so much similarity between primates is now dead with these new DNA findings. So let's see if there's a last slide, since this is not the usual ones. Oh yeah, here we go. So I assert that the DNA evidence for evolution is now incredible. It's kind of like the other fossil record. There's shared endogenous retroviruses among all the great apes. You can nest them in a hierarchy and show how, as we move farther away from chimps into orangutans and gorillas, that the similarities go down. There's human chromosome 2 fusion. We have shared pseudogenes. We have the gene for making egg yolk. Believe it or not, we actually still make parts of that, but we have a knocked out gene for making yolk, and because of our ancestors, and it's in the same homologous position as on the chromosomes that is in chickens. So it's great. There's great evidence for vitamin C production with a pseudogene for that and for other things. And then the banding of chromosomes you just saw. So we have in humans about 98.8% of the exact same of those 3 billion base pairs, the exact same ATCG sequences. And it's even higher in the protein genes that for producing genes. So check out my site. There's a lot of stuff that I think will be interesting, topics that are pertinent for today from a biological standpoint that I think you may find interesting. And sorry that I couldn't get my slides to work exactly, but thanks, Matt, for coming to the rescue. And hopefully I've left some time for questions. Thank you.
Yes, absolutely. You have left time for questions. And it's great. Let me see. I am having, can you hear me?
Yes, I can hear you well.
I am not getting, oh, I need to reset my self view on. Sorry. Give me just a second. But as I figure my stuff out, we do have some questions already in the Q&A tab. So you can use that Q&A feature on your Zoom toolbar to submit questions for John. There's a couple already in there. And there's one that, if I remember my biology correctly, my basic genetics, I think I know the answer, but I'll let you answer. So if two of 48 chromosomes fused, then there would be 47, not 46. So what's the answer to that question?
What happens is that if you shuffle the chromosomes around, you don't lose any DNA if you have a fused one. So people don't realize that there's something called a Robertsonian translocation, which actually occurs in 1 in 1,000 people. They're walking around with 45 instead of 46 chromosomes, and they don't know it because they haven't lost any DNA. When they find out is when they want to have children, and then you end up with a lot of miscarriage and Down syndrome and things like that. So if I'm answering your question right, sometime in our past, there was a fusion event, and it looks like it happened after we split from chimps. And so we went from 48 to 47. And then if you remember your biology, there's something called a Punnett square, where you can put the eggs on the side and the sperm on the top and take 23 and 22. Anyway, if you do that, it turns out that you can crossbreed, go back to somebody who's inbreeding who has 47 and somebody who has 48 chromosomes, and you'll make a 46 25% of the time. And then if you get 2 people with 46, they will have children that are normal at 46. So basically, this human chromosome 2 fusion, when it occurred, only took 2 generations to produce an individual with a pair with 46. And then for reasons we're not sure, it just got fixed into the population. I also found a book that was published in 1991 that had a diagram of all the fissions and fusions of chromosomes going back to 42. At that time in the 1980s, they didn't have the technology we have now. They were just using inversions of the chromosomes. So when you look at the chromosomes and it goes through recombination and meiosis, a piece of the chromosome can flip around so it's in the opposite direction. The body will still read it okay, but if you compare those inversions, those sections that have flipped with chimps and orangutans and gorillas, you can actually come up with a phylogenetic tree just using that, showing that in the past we've had a number of fissions and fusions of chromosomes. This is not a unique situation. It's just that it's the most recent one to us.
All right, thank you for that answer. We have another one. Since the prime example of irreducible complexity in the ID trial of Kitzmiller v. Dover was the bacterial flagellum, have you considered demonstrating how that evolved, or using that example?
That's been addressed after he wrote his book, Darwin's Black Box, and did things like that. And so so basically, Dr. Behe is one of the fellows in the Discovery Institute for Intelligent Design in Seattle, and he also has a position as a biochemist, I think, in Lehigh University. But his position in his latest book too, he just wrote another book coming up with more examples of irreducible complexity. And so his mission in life is to find something that can't be explained through natural processes. And so he had flagellum during the Dover trial, and also the clotting cascade. And so the problem is that or not the problem for him is that the flagellum could have evolved in other uses. So it didn't have to come from being a flagellum that just came out de novo. It was constructed from other things. And same thing with the clotting cascade. He said it's irreducible complex. If you take a piece out of this a very extensive clotting cascade for 10 factors that produce hemophilia, if you're missing one or two, it won't work. And then, of course, the biologist came up and said, well, actually, there's lots of clotting cascades in nature that only have part of what we have. And so it wasn't irreducible complex. And that's basically in terms of Behe. I haven't looked into all the steps that, especially Kenneth Miller has a video on how you can have a mouse trap and it can be used for other things. It doesn't need to have all the parts because it can be used for other things before it kind of evolves into a mouse trap. And so that's the principle that he's ignoring, that Dr. Behe is ignoring, is that that irreducible complexity so far has not been shown. But they just keep, it's playing whack-a-mole. They will keep moving the goalposts. As soon as you show that flagellum is not irreducibly complex, or the clotting cascade is not irreducibly complex, they move into other things. And so in his latest book, it's magnet for helping the bacteria line up and the eye. And so it's never ending. They'll just keep looking for more examples and we'll keep beating them down.
Well, related question to just that point. As genome sequencing and research advances, do more religious, intelligent design scientists begin to change their positions, or are they just looking for arguments?
No. As I mentioned, their origin narrative is crucial to they have to have a historical Adam and Eve. They have to have a fall because that's how sin came into the world, and that's why there's a need for a savior. So if there's no Adam and Eve, there's no fall, there's no sin, there's no need for Jesus. And they will fight tooth and nail against human evolution, except for one type of creationist, and that's the theistic evolutionist, or as they're calling themselves now, evolutionary creationists. Biologos and Francis Collins is that kind of member of that group. But despite Francis Collins, Dr. Collins, trying to get the word out to his fellow evangelicals that this DNA evidence is so sound and so solid that to continue to deny human evolution from a DNA standpoint alone is perverse, he's had very little impact, I think, on the old Earth creationists of Hugh what's his name, RTB, Reasons to Believe, and also, probably 85% of the advertisements or propaganda that's on the internet with creationism are the young Earth creationism, AIG and CMI, and of course Ken Ham. And he's with AIG, but ICR. So what you'll run up against most of, if the students are getting questions about evolution, it's normally going to come from the young Earth creationists, those three organizations. And they are not going to give it up. They can't afford to, because to them, it's a domino effect. And so we can, like I say, I've been doing this for 40 years, so I know their arguments and I know how to counter them if anybody wants to contact me. I will mention, however, that William Lane Craig, one of the greatest debaters for Christianity, just published a book. I think it was either 2019 or no, maybe it was 2021. And it's called In Quest of the Historical Adam. And thank goodness he finally admits human evolution, and he also dismisses most of the Pentateuch as being mythology. Yes, this is William Lane Craig saying this in his book, but he's got to keep a historical Adam. He's got to figure out how to get Adam and have his cake in human evolution too. And so it's an interesting book how he does it. So I'll leave that up to you. But you can't fight the science forever, but they are.
So a biologist friend of Jim's, who submitted the question, says life is inevitable in the universe. Is that possible?
I have read some articles by people, well, everybody's smarter than me usually in this area. I'm just putting them together, that says that there is something about nature that's emergent, that given time and the raw materials, the recipe, that life will keep emerging. But that's a question of abiogenesis, of producing life from non-life, and that's not evolution. Remember, evolution is the origin of species, and at this time we don't have a good scenario worked out on how life could arise from non-life. So I don't know. There's the RNA world hypothesis. They're making great progress. Actually, NASA is giving people grant money to find out how this could occur, because they're looking for life in the solar system, in the universe. But I'm just satisfied with, let's just talk about evolution, which is the origin of species, and let's contrast and compare different origin narratives to see what's true and not.
All right. Well, that brings us to time. Works out perfectly. Thank you so much, Dr. Peters. This was a very informative talk. I was telling Dr. Peters as we were preparing for this, especially the example of RNA viruses, retroviruses, and the impact those have had both on our own evolution and on traces, and being able to trace that is just, I'd never heard that. It was mind-blowing to me. And so thank you for that, for a very informative talk. We will be posting this to our YouTube channel, which will be linked on our webinars page. So you look in the chat, I put the link there, secularstudents.org/webinars. You'll be able to view that again and send that to your friends tomorrow. And then also, please check out those upcoming webinars, register for those. There's links on that webinars page to do that. Thank you all so much for being here. Thank you, Dr. Peters. And we'll see you at the next SSA webinar. Thank you.
Thanks.